Thank you, Nikolic and Catherine. It's a real pleasure to be invited to speak at this seminar series. I've looked at the previous speakers and I'm entirely flattered to be invited. What I'd like to do today actually is to address some key issues, but also share with you some of the data that was generated by my group at the Institute of Cancer Research when I was there. And really, some of it, but not all of it, was used as the basis of founding Monte Rosa Therapeutics, which is now based in Boston. One of the biggest challenges and actually opportunities in drug cancer discovery is that defining drivers of cancer and validating new targets still remains a big challenge. And even when we know the known drivers, it really has been difficult to drug most of the sort of leading drivers such as MYC, RAS, alternative splice forms of the androgen receptor, for example. And many of these are difficult because they have shallow pockets. We don't really understand a lot of drivers of cancer beyond the kinome, I would say. And when we do discover some of these drivers, there's limited published data, and it really does require a lot of target validation before you can actually commit yourself to investing, say, the millions of dollars that's necessary for cancer drug discovery, for example. And often, and this is a theme that I'll develop throughout this talk, is that getting targets that are unique to cancer, differentially expressed, is difficult, and increasingly we're realizing that cancer drug targets are context dependent. For example, deubiquitinase inhibitors, in some deubiquitinases are tumor suppressors in one context and drivers of cancers in the others. And I will put to you that possibly the best way moving forward now is to try and change our paradigm of cancer drug discovery and not just rely on high throughput screening and bring in more predictable models and bring in phenotypic screens earlier, given the fact that we can now deconvolute these targets. And this, at least in part, will be the theme of my talk. So most of my talk really is going to focus on what lessons we've learned so far from targeting cereblon. Can we extend cereblon binding enhancers of protein-protein interactions to solid tumors? So far, their work undertaken at Celgene, but now increasingly by other groups. When it's come to cereblon in terms of molecular glues of protein-protein interaction enhancers, the success story has been in hematological cancers, but can we find chemical moieties and approaches for solid tumors? And I will share some data with you in that context. The second aspect of what I'll share with you is actually using the imide-associated degram, which largely has been described by the group here in Boston and Nico Thomas' group in Basel. How can we exploit that for tagging target proteins to understand protein function and target validation? And then I will show less data, but also give you some of my thoughts about how we should be thinking about moving beyond cereblon and what other E3 ligases would be fruitful. But the whole underlying theme of my talk is really, and this is the whole concept, I think, about targeted protein degradation, is to actually bring proteins together and enhance, and then subsequently enhance the degradation in the right cell context. And throughout the time I've been involved in this, the thing that struck me the most is that every time we do experiments with the right cell biology, we uncover new biology, new pathways, and new contexts in a way that genetics would not do, because we really are interrogating the proteome in a very different and structured manner. So the story, is all of you know, is that as a consequence of the discovery of cereblon as a target for thalidomide by Hiroshi Handa's group in Tokyo, a number of groups, and one of the things that humbles me most is when I'm presenting you a lot of the data here, it really has been undertaken by the group here in Boston. And I just have to say that when I'm presenting some of my data, it appears that I'm entering a lion's den. But nonetheless, the story, as you all know, is that it was very clear very soon after that discovery from Tokyo that what the imids and thalidomide analogs were doing was they were binding cereblon and really recruiting neo-substrates, which normally would not be degraded by this Cullen E3 ligase, for degradation and presentation to the proteasome. And in the context of multiple myeloma, it's very clear that there was degradation of T transcription factors, zinc-finger transcription factors, Ikaros and Aiolos, and their consequent degradation led to a decrease in what is called the hallmark of multiple myeloma transcription factor called IRF4, and subsequently decrease in c-Myc. Now, what I want to really point out is, quite apart from this discovery of neo-substrates, one of the most intriguing factor to me certainly was that the Ikaros and Aiolos had not been described as potential targets in multiple myeloma. And the one lesson I've learned from this is that this really clearly shows that multiple myeloma is a B-cell developmental problem and not just a proliferation of terminally differentiated plasma cells. And that has got implications as we think about resistance to some of these pathways, but also other novel therapies, including new degraders. So what have we learned so far from targeting cereblon? So the early drugs, lenalidomide, which were in the clinic, clearly shown, as I've already described, in the context of multiple myeloma, to degrade Aiolos and Ikaros, and actually lead to not only the killing of multiple myeloma cells, but actually degradation of Aiolos and Ikaros, we've shown, and a number of other groups have also shown, it also activates T cells. And actually, it's this dual activity of killing B cells by activating T cells, which really give the IMiD agents a very unique profile of activity in this condition. And I also want to make the point is that, largely speaking, a lot of people have worked on substrates of consequence in cancer cells and hematological malignancies directly on the malignant cells, but less work has actually been undertaken on how these agents can modulate immune cells, and I think that's in the future is going to be a very fruitful area for further investigation. But the other thing, and this was work Celgene did in collaboration with Ben's Ebert's group, was to show that lenalidomide not only degraded Aiolos and Ikaros, but also degrades casein kinase 1 alpha, and at least in part explains the activity of lenalidomide in 5q minus MDS. But the next thing was, if you look at the chemical structure of the IMiD agents, they bind into the tritryptophan cage of cereblon with the glutamide interacting with the tritryptophan cage. And the next obvious thing was to extend or change the structure of the thalamid ring to see whether you alter the activity of this the IMiD agents, as they were called initially and now subsequently called cell mods, and by extending the thalamid ring, it's very clear that this compound CC885 was able not only to degrade Ikaros, but also degrade GSPT1, which is translational termination factor, which I'll talk about in a minute, and therefore you could extend the activity of these IMiD agents. But what was very clear is that these agents were very much cell context dependent, because lenalidomide degrades, for example, casein kinase 1 alpha, but pomalidomide, which is a closely related compound, does not degrade casein kinase 1 alpha. So it's not just cell context dependent, but subtle changes in chemistry can alter substrate profile. And again, every time we do these experiments, we target the new biology, because the role of casein kinase 1 alpha had been speculated upon, but and this was really confirmation that it had played an important role in MDS, including all the animal studies that we've done in Boston. The other things that we've learned is that the target degron contains C2H2 domains within the zinc finger transcription factors, and of course we're also degrading non-zinc fingers. The key thing for both of these is that it's the necessity of the stabilized beta loop, stabilized by a sentinel glycine, which is critical for degrading any targets, and that the degron sequence interacts both with the compound and the substrate receptor. And substituting or altering the thalamid ring, as I've said and shown you for 885, alters substrate specificity and also the range of cancer activity. So lenalidomide, for example, did not have activity in acute myeloid leukemia, but CC885 at that time was shown very clearly to have activity in acute myeloid leukemia. And important to point out, limited activity in multiple myeloma. The other thing that really is emerging is that the affinity of the binding substrates to the drug cereblon complex does not always predict for degradation, and that's where cell context dependency comes in. It's the relative abundance of the substrates, but also availability of these substrates for degradation that is really important. And therefore the concentration and the expression in the cells, and which compensates some of these target proteins exist, is something that is going to emerge as we undertake further experiments. And a lot of these substrates are often in epistasis and in competition for cereblon binding. So this is all the work done largely at the Boston group. And when I moved from Celgene back to the UK, one of the key challenges was how could we use some of this information to really think about undertaking experiments to target solid tumors as opposed to hitherto the activity in liquid tumors. And I'll really give you a story that has emerged and some of our thinking that we started off by thinking. The first thing we really wanted to do was create models which really as closely defined the targets that we're going for. And one of the observations we had early on at Celgene, and it wasn't fully exploited, was that whilst the IMiD agents were great at targeting liquid cultures in non-3D states, we've got hints that we were seeing activity of some of these agents when we did 3D cultures. So very early on, the idea was to not only create cell context models, but also to use 3D models to see whether we could tease out activity of these agents in that context. And there's a whole story around that. I will not talk about it, but there is often differential activity of some of these agents when you put them in spheroids as opposed to conventional liquid cultures. And so creating the right cell context was important. The second thing really was to create a set of chemical probes with the correct molecular weight, the right log D and permeability with all the pharmaceutical properties so that we could extend the library beyond what existed and actually use that to explore near substrates if we got activity in solid tumors. And the hope was that all these near substrates would be degrons containing substrates, but we weren't sure at that time whether there'd be other targets which did not contain the degron. But the key thing that we realized earlier on is that when we use cellular systems, that when we do find compounds in an extended library known to bind cereblon, that they have very tight SAR. In other words, there's limited scope for further chemical optimization. So if you get permeability with a certain compound and then you alter chemical structure, you just by changing certain motifs, you alter the pharmaceutical properties. In other words, it's a very tight SAR. And I'd like to just really make the point that this approach was empirical, is using biology and cereblon as a template for cancer drug discovery, for understanding new biology. And it's very distinct from PROTACs, which really need to define the target, and often because of the linkers, some of those compounds can have poor drug-like properties, although, as you well know, a number of PROTACs have now been optimized for ADME properties. But, and I would argue so far, many of the conclusions that we still are getting is based on empirical studies and largely from phenotypic screens. In fact, the history of Celgene has been a history of phenotypic screens, and that has taken at least 4 to 5 compounds to the clinic, and 2 or 3 which are blockbusters. So the problem we wanted to tackle is the problem in solid tumor cancer biology is, can we target MYC? And the key consideration, as I've already said, is getting the right cellular model. And we wanted to create a cell line model which is dependent on MYC, okay, and was vulnerable to MYC, so that when you took away MYC, the cells died. And essentially, we wanted to also recapitulate what happens in cancer. When you get your first hit in cancer, and MYC is very much a truncal alteration, the amplification of it, an early event in cancers, and over time, cancers adapt to MYC. We wanted to really try and identify the modifications and adaptations in the proteome and potentially the transcriptome over a period of time. So that was the idea of developing a MYC model. Subsequently, I think this approach should and can be used for RAS and all the other difficult to drug models. So the model that we decided to use was human mammary epithelial cells, which in themselves are not malignant. So if you inject them into a mammary pad of the mouse, they will not grow. However, if you transform these with c-MYC, they will actually show activity, as I'll show you in a minute. And so what we did was to create 2 models. One model was a tetracycline-induced c-MYC expression in human mammary epithelial cells with a GFP tag. And as a control, we put a P2A skip with GFP, so we uncoupled GFP from c-MYC. And that was just a way of making sure that we didn't interfere with c-MYC activity when we were looking at our models. And really what we were saying was that we're going to put a cereblon binding library. It would actually be the chemical equivalent of RNAi, and therefore we would define proteins that were adapted to c-MYC and were synthetic lethal partners of c-MYC in the context of human mammary epithelial cells. Another story, we also engineered and knocked in EGFP into the endogenous locus of MYC, and actually then mutated the T53A site, which actually degrades MYC through F-box proteins, as a way of looking to see what happens when you activate MYC under the control of an endogenous promoter. And I won't talk about this, but we got very different biology built. We wanted to develop this model to really look at the proteins that develop c-MYC and are also synthetically lethal partners. But I'll only talk about the first part of this model. And so the first thing was to overexpress MYC GFP, and then to do a doxycycline titration curve. And you can see very clearly you get a dose response increase in MYC expression. And most importantly, we have controls. So you have your HMAC parental controls, which really shown here. You have the HMACs with EGFP without MYC. And when you add doxycycline, you maintain viability, but these cells do not grow and proliferate. However, when you switch on MYC per se, you get increase in these 3D spheroid cultures. Remember, I said we were going to use 3D spheroid cultures. And not only did you get increase in the spheroids, but you see these mycelia coming up, which is really showing this invasive propensity of driven by MYC in the context of 3D culture. So we've tried to reproduce as closely as possible in a 3D culture system a model of MYC, and I'll come back to the adaptation aspect later on. But to show that these cells were truly dependent on MYC, we use what Gerard Evans has described as the hallmark of MYC activation, is that when you take MYC cells and you serum starve them, and we now understand why, because MYC's propensity to drive cancer metabolism is that you actually cause apoptosis. And you can just see here in these adapted cells, when we put them in 0.1% fecal bovine serum, they actually die. So this gave us confidence that we had adaptation. And in order to get adaptation, really found something quite interesting, and this is based on Trey Westbrook's work at Baylor College of Medicine. It showed in a very seminal paper in Science about 10 years ago, is that when you overexpress MYC in HMAC cells, they don't immediately become dependent on MYC. You've got to put them through a number of population doublings. And what we found was that if you took these HMAC cells and put them through 25 population doublings, that's when you got MYC dependency. And so that's where we did the experiment. We took the HMAC cells with GFP and MYC linked to GFP with a P2A skip. So you did interfere with the MYC activity, and then you plated them with doxycycline in 3D culture, look for endpoints, both for steroid size, EGFP fluorescence, and cell death. And most importantly, it did them at different time points. And we then exposed them and screened them with a novel cereblon binding library that was developed by Ian Collins, my colleague at the Institute of Cancer Research. And what we found very clearly, and this is, we used CC805 as a control, but we found very nicely is that we were getting activity of the MYC. These are MYC-driven cells in the front. I can't with the pointer. Here, these are MYC GFP after exposure. After 5 days, you can see that all these cells have died, and the GFP controls, the normal HMAC cells, are still alive. So we got a differential killing of the cells with these two compounds, and all of these, because we'd used cellular systems, had good pharmaceutical properties, as I'll show you in a minute. So the next question was, what is the substrate of consequence? And we did, like everyone else, TMT proteomics, and we found that the adapted protein was CMYC, that was GSPT1. So in other words, GSPT1 was the synthetic lethal partner for MYC-activated cells, and using hybrid assays showed very clearly that GSPT1 degradation was taking place in a time-dependent manner as well as a dose-dependent manner. We also then took some of these compounds and screened them against a variety of triple-negative breast cancer cell lines, and the triple-negative breast cancer cell lines that were MYC amplified showed exquisite activity for these compounds. And to further show that GSPT1 was the neo-substrate of consequence, it's a very clean mass spec, but to just further confirm it, we actually use a dominant negative GSPT1 motif where we mutated the sentinel glycine, and you can see very clearly that the mutated sentinel glycine showed no activity in context, whereas the wild type did. So the phenotype that we'd shown was absolutely dependent on GSPT1. And as I told you, this compound had good pharmaceutical properties, and we also showed activity in a triple-negative breast cancer cell line model in vivo. So this really, although GSPT1 has been identified as a target in acute myeloid leukemia, there was no link between this and the driver and the link between solid tumors, and subsequently also shown that other MEK-amplified solid cancers have got activity for these novel compounds. And at least in part, some of this work is linked to the founding of Monte Rosa, and Monte Rosa is going into clinical trials really targeting GSPT1 in a range of solid tumors, including small cell lung cancer. And actually, this data has more recently been confirmed by the St. Jude's, where they also showed chemical synthetic lethality in a variety of MEK-amplified cell lines, which were medulloblastoma, but also hematopoietic cell lines, an AML cell line in this case, and shows very clearly that they've also got compounds that degrade GSPT1, and that phenotype can be rescued with a similar dominant negative GSPT1 construct. So this, at least in part, shows very clearly that GSPT1 can be targeted in solid tumors, but also gives you a marker that c-MEK amplification may be a way of selecting patients for therapy. Now, this is quite a surprising thing in many ways, in that GSPT1 is regarded as a house keeping protein in some ways. It has got critical functions initially described as G1 to S phase protein. GTPase is important for G1 to S phase transition, but then subsequently largely work done in yeast, showing that it associates with the ERF1 to mediate stop codon recognition and release of nascent proteins from the ribosome. And also importantly, and we shouldn't forget that, it's really important for the main types of mRNA decay. And so the hypothesis actually that comes out of the cell gene group is that when you degrade GSPT1, you really lead to effete proteins being produced, release to activation of the ER stress pathways, and this leads to apoptosis. Now, one point to make is that MEK has really been shown not only to amplify transcription, but increasing data is that its major activity may be at the mRNA and translational level. And the new biology I would say here is that MEK threshold of translation, termination, and mRNA decay in the amplified cells, and that's why you get the differential effects between normal and MEK amplification to give you the therapeutic index that's necessary to go into the clinic. And I think this is a really important finding, is that these so-called critical genes for housekeeping or important cellular physiological activities can still be targetable because the thresholds have changed when we've got oncogenesis going on. And again, it's not always the oncogenic driver that's important, but maybe the synthetic lethal partner that's will give us opportunities for cancer drug discovery, given the fact that a lot of the known drivers are difficult to drug. And I'm not saying this is, and that's something that we should keep in mind. The other thing I just want to point out, and I ask you to read this paper in the interest of time, this come out of the cell gene group, which suggests that the activity of GSPT1 is dependent on the expression of cereblon based on these splicing factors, alternative splicing factors ILF2 and 3, as well as the mTOR pathway. I can tell you that in the c-MEK solid tumors, I don't think this is the entire story. I think there's a separate pathway here. So there's other discoveries to be made in that context. So the question, in one way, we've got GSPT1 is degraded. and we see a path forward to the clinic, and that is exciting because we linked an oncogene directly to a pathological pathway and a therapeutic paradigm. However, in some ways, it was disappointing, I'd say, that we picked up GSPT1. And the question that always comes back to mind is that, are we in a closed loop? If we're going to use cereblon, are we just going to get these zinc fingers, casein kinase 1 alpha, GSPT1? Or as we change the cell context, as we change the chemistry, that we will get other exciting targets? I think time will tell, but that, I think, is the key question, is can we move beyond this empirical approach that I've described to be able to create drugs that are targetable by design, which is what the Protacs do in that context. And, you know, everyone's got their favorite way of looking at these G loops, and this is where at the computation biology, my colleague Bizan Lazikani did at ICI, she's now moving to MD Anderson, suggesting that, and using a database called CANSAR, suggesting that there are at least 5 strongly validated G loop containing proteins which are not largely in the public domain, but then there were 58 that potentially could be, and then 84 that have these structures but no published data on. So there's a rich theme still to be mined here. The question is, can we design our chemistry and our cell line models to really pick these up in context? And at the moment, I don't think we've done enough cell line context biology in the way that I've described. I think if we're going to make progress, we need non-glutaramide binders to the tritryptophan pocket. Uracil is known to be binders, but I think we need other moieties. Can we find them? Can we substitute the lethalimid? And I think it's getting all that with structural biology information that will be important. But once again, I just want to make the point that we've got very tight SAR here, and subtle changes really alter both pharmaceutical properties, substrates, but also potential side effects. So in the context of drug discovery, I don't think we're quite there yet, but undertaking these patient, thoughtful experiments across the chemical space with the appropriate cell line models will, I think, be a rich theme still to mine. So the second part of my talk, which I'll speeden up, really is to exploit understanding of the image-associated degron for studying protein function and target validation. And really here, and again, I feel a bit humble talking about degradable tags with this group, but they are key things that one needs for tagging for studying protein function. Can you get rapid degradation over a period of time of hours? Has it got catalytic mechanism? And most importantly, does it specifically remove all your protein functions so that it acts as a switchable chemical mimic of RNAi? And basically, the inspiration for trying to exploit this system was really the DTAC system that obviously was discovered at the Broad, which really is a PROTAC, which really targets cereblon on one end with the imide-like agent and binds to the mutant FKBP12 fusion protein, which is tagged to the protein of interest. So the question we asked was, could we use and find a minimal degron that we could bind to a target of protein of interest, use EGFP plus that degron, and we called it a degron containing domain, DCD, and use that as a way of degrading. And again, we've used some of the information that came out of the group here in Boston to see whether we can find the right sequence and keep a minimal sequence that has got acute activity, degrades as many proteins as possible to almost zero, and most importantly, and regulated using a tetracycline system. And to design this, we actually designed 23 DCD tags, as we call them, with different ranges of amino acids. And again, this is based on the structural data from Nico Thomas' group, as well as group in Boston and Celgene. And we really got these tags based on GSPT1, ICROS, and other zinc fingers. And we basically had 3 groups: beta-hidden loops containing GSPT1, casein kinase 1 alpha. And I can tell you these are not efficient tags at all. And so the second group was really zinc fingers based on ICROS and Iolus. And the third group, and it's very close to the tag that Ben Ebers has also described, using really a chimeric zinc domain from ICROS instead of P91, which was based on that paper that came out of Nico Thomas' group as well. So what we did was to create a doxycycline-induced system, and we used MYC as a first proof of concept. We used EGFP attached to this degron-containing domain, and as a control, we also put EGFP with P2A skip. And you can see that when we add doxycycline, when we add doxycycline to the system, we switch on MYC and EGFP, and then, and then when we add CC885 as a degrader, you literally degrade your target protein, in this case MYC, and you get a decrease in fluorescence. And as a control with the P2A skip, you don't have any activity in context. So we use these 23 different degrons and look for efficiency of degradation, reproducibility and robustness, and no drug-induced toxicity. And I just want to say at the get-go, CC885 is not the right image to use, because we use a whole range of images, and the most optimal, to cut the long story short, was the sequence DCD23, which is the chimeric construct of ICROS and ZFB91, 60 amino acids, and they're 7 kilodalton size. And the optimal partner for the degrader was CC220 in context. So we used this system, and we thought that we'd do a head-to-head comparison with the DTAG system. And there were 2 clear differences: is that the DCD23 was a rapid degrader within minutes, but equally importantly, we did not see the hook effect that we do see with the DTAG system, which again, if my pointer will help me, when we're targeting, for example, RBM39, you do get degradation, but then as you increase the dose of your drug, you get the hook effect. And we do not see that hook effect at all with the DCD for, in this case, RBM39. But we've then gone on, in collaboration with Trey Westbrook at Baylor College, really to look at degradation of a wide range of proteins. I've shown you the data on RBM39, looked at CDK9, looked at spermidine synthetase, and we've also looked at the interferon stress pathway genes, MAVs, and consistently we degrade almost completely our targets, and we do that rapidly, and once again without a hook effect. So and then we went on to show in vivo that when we transduce HMEC memory cells with EGFP, with the DCD23 degron, within minutes of treatment, we get degradation of the target gene, and that occurs at 4 hours. And you've also got the P2A skip, where you do not see any degradation. And basically, this really is validation that the system not only leads to potent loss of fluorescence in these targeted genes, but that occurs within 4 hours. And this was one single treatment of CC220. And this are the models which is targeting a human cell line model in the mouse, really is quite good because CC220 will not cause side effects in the mouse background. So what we've got really is quite a robust but simple tag. It's of a size that hopefully will not interfere with protein function, and we're now done C-terminal, N-terminal tagging. It's specific, and that specificity is really provided by having relevant and corresponding P2A controls. And CC220 was the optimal IMID, and we are submitting this for publication, and all these will be openly source available for use by the community. So the third part of my talk is less data and more really now thinking about what we're going to do beyond cereblon, or is cereblon still an area that we should continue to explore? My personal view is that I still think there's a lot more to do with cereblon, and a number of questions come to mind. Mostly, is there yet another pocket on cereblon that one can exploit? I think that's going to be an interesting one. And then what other E3 ligases can we target? And there's already been some great progress in the last couple of years within that context. But my whole premise is that whatever we do, we should not just rely on chemistry. We should bring in structural biology, which we are doing, but also bring in appropriate cellular models for disease context, because it is it's the integration of all of that plus computational biology, because a lot of information comes out of that, which will lead to the success. And this really is the problem posed by Ivan Dikic, really, and his colleague in a recent review, which is, what do we do beyond the existing E3 ligases? It makes the point less than 2% of E3 ligases have been exploited for target protein degradation. And if one looks at the distribution of potential targets, receptors, for example, the decals for the Cullen family or other E3 ligases, then this is the range of challenge. And the range of challenge, and he just brings this point about tissue specificity, is that there's different ranges of expression, and therefore, if we are going to get this right, it's going to be a patient journey of defining both the biology, the expression, finding chemical probes that bind these potential receptors or E3 ligases, and then doing patient cell context and disease context experiments, not only to find substrates, but also to translate that into the clinic. And I will put it to you that this sort of work will be done by enhancers of protein-protein interaction, which will be largely molecular glues, because there we will be using libraries to understand that in the way that we've done hitherto around cereblon. The question I would also then bring in to say that cereblon, being a decaf, is one of 60 decafs. And even before we start thinking about going beyond the Cullen 4 family, I think we've got great opportunity just within the decaf family. And largely for a couple of reasons. Again, I think they have certain features. A lot of them are ubiquitously expressed, but we again need to do patient immunohistochemistry, get the right antibodies, reagents. A lot of them are nuclear and cytosolic. Some of them do have structures, and in fact, some of them can be targetable by design because they've got WT40 domains. But I will still continue to say that we need to do the cell biology experiments. But the other question, this would be interesting for discussion, is the endogenous substrates for a lot of these are not just defined. But if we get the right chemistry, I think the phenotypic consequence is going to be dominated by the neo-substrate and not necessarily by displacement or endogenous substrate. And that's so far the story of with cereblon. And so, but ultimately, whatever we target, they've got to be as close to cereblon in that when we target them, they've got to be functionally neutral. So that's the sort of patient work we need to do. But I do think the decafs in the Cullen family are important, largely because you know, the flexibility of the Cul4 ligase arm, but also the surface area that brings your E2 ligase bound to RBX to contact your substrates of consequence is such large that I think you can bring into play larger protein and protein-protein complexes in order to effect degradation. So if I were to put my money on the next generation of targets, it's going to be the decafs within the Cullen family. Now the decaf 15 largely degrades alternative splicing targets, RBM39, and maybe it's limited. I do think the other decafs are going to be important, and understand and the fact that many of them are overexpressed in different cancers really gives you a therapeutic index as well in context. So I would say that there are a number of potential targets, and this is something that I'd be very interested in pursuing. Now, that is not to say that the other E3 ligases, as have already been, such as for the Cullen-3, the elongins, are not going to be a useful approach as well. And then the work from Danny Numura's lab about ring fingers is also very exciting in context. So I'll leave this here to say that there's a lot to do, there's a lot to think about, but maybe I'm biased, but don't forget cell biology. So I'll leave it there. I'm happy to take any questions. Before I do that, I'd just like to acknowledge some of my colleagues in person. So I'd like to acknowledge I've already done Ian Collins and John Caldwell for the chemistry. Person I'd like to really acknowledge are two individuals. This Hannah Wang, who single-handedly did the HMEC model, is a torch, is a force of nature, and Habib Berganina, who's smiling at the back here, let me point him out, did all the work, largely most of the work on the DCDs, and I'd like to acknowledge his major contribution. And I'm happy to take questions.